From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on February 20th, 2026. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 125th episode of Matters Microbial. Thanks for every guest, listener, and viewer. Please spread the good microbial word far and wide. As most of you have seen and heard, I adore working with bioluminescent bacteria. Seen here is a fine streak plate showing luminous photobacterium. It brings me such joy. My wife, Dr. Jennifer Quinn, and I often use this bacterium to create artistic Lex portraits, like this one of the late Carl Woese, seen here. Woese is a name every micronaut should know, and I'll put a link in the show notes for you to enjoy. Bioluminescent bacteria require oxygen to produce light, so I had our department engineer create this rocker tube seen here in the light. I poured in a luminous culture of bacteria, turned off the lab lights, turned on the rocker, and filmed the result with my phone, as you can see in this short video. The light follows the bubble of air in the tube. It's a Zen moment to watch. As you will recall, my research students and I are exploring microbes that live in reusable water bottles. We call them water bottle buddies, as seen on this sticker. When we do this kind of work, the goal is to isolate single colonies from our first swabs, purify them, and do single colony PCR to amplify and sequence the 16S ribosomal RNA gene, which is something like a barcode. Over the years, we've isolated many types of water bottle buddies, seen here in this composite photograph. It kind of is a rich ecosystem. My students would write short reports on their water bottle buddy, like the one here. We generally find bacteria that are biofilm formers and are able to adapt to low nutrient conditions. I even put together a word cloud, seen here, of the types of water bottle buddies that we've seen over the years. The problem is that not every bacterium swabbed will grow in the nutrient medium provided. We've only seen the ones that grow on R2A medium. R2A medium, by the way, is a medium designed for growing things that are found in water. It's often used in water quality studies. Micronaut Chiana swabbed the straw of her water bottle, and there was visible material on the swab, seen here. Swabbed onto an R2A plate, seen here, Chiana had a rich population in her water bottle straw. I was able to find a vendor who could determine the bacteria present without cultivation through high-throughput DNA sequencing. It was a little bit more expensive, but just look at the results from Chiana's water bottle, seen here. You'll see the abundances of types of microbes found out of 5,000 reads of 16S genes present. Thus, the top 20 water bottle buddies in Chiana's water bottle without cultivation. For example, 11% of the reads were from Pharyngiumbacter, a gliding bacterium often found in fresh water. It does make biofilm, so this makes sense, but I have never observed it from a student colony isolate. If you look carefully, you will see some of the microbes in the word cloud appear in the abundance chart, like Chrysiobacterium and Brevidomonas. Wow. Now, my students have isolated these many times from water bottles, so it's nice to have seen it in the high throughput results. I also decided to swab the spigot of our deionized water system we use to make solutions, seen here. It's just water, right? Not so much, friends. Swabbing onto an R2A plate, seen here, shows a fair amount of bacterial growth. But the non-cultivated results are fascinating. Seen here are the abundances of microbes found from, again, 5016S ribosomal RNA reads. 41% of the reads are from Pelomonas. Now, this organism is often found in mud, so I don't understand that. But I would argue that the community found in the water spigot are mostly two taxa. Now, are splashes providing the population in the spigot? Or do they arrive on dust? I don't know. All of us move through a cloud of microbes at all times, after all, and despite the media reports, most microbes could care less about us, what I call meh microbes, as opposed to harmful or helpful microbes. After all, they have their own agenda, and it's not ours. Currently, my goal is to look at several water bottles, as described before, to see if there are differences between the microbes found in metal or plastic water bottles. Also, are there differences between different water bottles of the same type, or is there a core community? This is very different from the way I've explored water bottle buddies in the past, and I find it exciting. OMG, overwhelming microbial greatness for certain. I hope to find patterns. It's also a great way to get students thinking microbiologically. After all, water bottles or any surface are really microbial ecosystems. I'll keep you all posted, and how I wish I had grant money for this kind of analysis. Moving on to today's topic, I want to remind listeners and viewers of the importance of antibiotics in our modern world and the threat of antibiotic resistance. This is something that ought to be in every single class that we teach in biology because it's critical to so many things. Streptomyces and related microbes are fascinating organisms. They have a complex developmental cycle, and many make antibiotics. They also make a chemical compound called geosmin, which actually smells like potting soil. But to be honest, the reverse is true. Please enjoy this short video from one of my prior microbiology labs after a student smelled a plate of Streptomyces. Enjoy.
That smells like dirt.
No, dirt smells like Streptomyces.
I spend a fair amount of time in my microbiology classes discussing antibiotics and the challenges surrounding them. So I am so very happy to introduce Professor Paul Hoskisson of the University of Strathclyde in Glasgow, Scotland, to discuss the work his laboratory and colleagues have done studying how bacteria like Streptomyces develop, how they make antibiotics, and how they can evolve even in the laboratory. Welcome to the Quality Quorum, Paul. We've communicated quite a bit over the years in social media, and I'm delighted to electronically meet you.
Hi, Mark. It's an absolute pleasure to be here. Thanks for the invitation. Nothing pleases me more than talking about Streptomyces.
I almost did a postdoc in this area. I hate to tell you, but I love all the different developmental mutants, but we won't get into that. Otherwise, it's just micro nerd mania.
Yeah, I mean, one of my postdocs was on exactly that, mapping and trying to find new mutations in development, and there's still many out there to be discovered.
And I think it's interesting because we tend to forget how complex the soil is. And I know you can find the actinobacteria in other places in the soil, but they are actual chemists. Before we get too deep in the weeds, I think for listeners and viewers, it might be really good if you could list some commonly used antibiotics that are derived from Streptomyces and relatives that perhaps people have heard of.
Okay, so, I mean, the conventional number that we always throw out there is about 2/3 of all clinically used antibiotics come from Streptomyces. I think it's probably a little bit lower, mainly due to taxonomists splitting a whole range of organisms out of the genus Streptomyces, but more broadly in Streptomyces and their relatives, it's about 2/3, and that is things like tetracycline, oxytetracycline, which is used as a precursor to make doxycycline. Erythromycin is made by Saccharopolyspora, which used to be referred to as a streptomycete. Clavulanic acid, kefamycin C. Let me think of others that even we talked a little bit about things like rapamycin as immunosuppressives, and immunotherapies. I mean, there are just so many of these molecules that spring to mind.
And we're not even using the trade names.
No, mainly because I can't remember all of the trade names.
It's absolutely true that we owe a great deal of the antibiotic revolution to one genus of microbe.
I mean, I forgot about streptomycin, which was the first of the kind of major antibiotics that were discovered from these organisms. That was the first antibiotic that was used against TB. So, yeah, huge numbers of molecules that people will know and have probably taken. Chloramphenicol is used for eye infections. Less commonly used now is kanamycin and the semi-synthetic derivatives of that, like amikacin. But these molecules are everywhere in the environment, and we use them all over the clinic as well.
So how about giving, and again, sometimes I bring this up, this term thumbnail sketch always makes me wonder, is it from art? As a couple of people suggested, who knows? How about a thumbnail sketch of the Streptomyces?
Great. So I actually work in a pharmacy department, so I always like to remind my colleagues that Streptomyces are much more talented pharmacists than they can ever be. So Streptomyces, I like to refer to it as nature's pharmacist, and you can see on the slide some of the beautiful images of Streptomyces, and also highlights some of the amazing biology that these organisms do. They're gram-positive saprophytic organisms. I mean, they can be isolated from pretty much every gram of soil there is on the planet. They've been isolated from permafrosts in Antarctica right the way up to the highest peaks in the Himalayas and deep sea environments. So they're pretty much everywhere. They're unusual for bacteria because they have large linear genomes. So most bacterial chromosomes are circular. And the average kind of E. coli genome is 4.5 megabases. In Streptomyces, they can be 8 to 10 megabases. So that's 8 to 10 million base pairs, encoding an absolute wealth of catabolic and metabolic function within those cells. I think they're probably best known for their antibiotic production, but that's intimately linked to their amazing developmental life cycle as well. So they start life as a spore, and that spore germinates and grows apically from the tip. So the new material for growing the cell wall is actually added at the tip of the extended hyphae. And because these are non-motile organisms, they forage through the soil, releasing hydrolytic enzymes, breaking down complex carbon sources in the environment. Their metabolism is actually really closely geared towards taking up things like N-acetylglucosamine, which is a breakdown product of chitin, which, as you know, is a major component of fungal and insect exoskeletons and fungal cell walls. And they form this mat of mycelium within the soil. And then when they get stressed or they sense that they're starting to run out of nutrients, they can erect aerial hyphae. And there's a whole kind of complex of molecules that they produce in order to break the surface tension. Because if you're a very small bacterium, the actual tension within the liquid air interface is actually really quite a difficult barrier to break. So they use hydrophobins, like many of the fungi do, to break this surface tension and grow up into the air. They then curl and they lay down sporulation septa, which allow the spores to then form within the sporulation septa, and they lay down about 100 of these sporulation septa simultaneously, which then mature into unigenomic spores that can go off and start a new colony of Streptomyces in a more favorable environment.
They're fascinating beasties, and one of the things that always amazed me, and I saw this with cyanobacteria, is sometimes the nomenclature used is reminiscent of things that are not bacteria. So you hear things like, I don't have a problem with spores, but hyphae, and that's what they look like. I've heard people talk about sporangia, all kinds of things of this nature, and this is because based on the appearance. What's interesting to me is how they kind of burrow into media, and then on top you can see the spores starting to form. So you get these, as you have shown, these wonderful photographs of the colonies. I can always tell an actinobacterium on a plate. I have found them certainly when I've looked for antibiotic producers in the soil. I see them in lichens and I see them in mosses as well. But It's funny, and you'll smile about this. My students hate them because they're hard to pick up with a loop.
Yep. Yeah, they often have this waxy appearance. So you can actually see again on the slide is that when they're forming this mat of vegetative mycelium, they're very waxy, almost like a standard bacterial colony. And then they get this powdery appearance as they start to initiate development. And you can see how hydrophobic the surface of the developing colony is in that image, where you can see that beautiful blue antibiotic called actinirodin sitting as droplets on the surface of the colony. This is actually a mutant, which kind of allows us to really highlight that it's this initiation of development that they also start to make this plethora of natural products that have antibiotic, antifungal, anti-helminth, immunosuppressive, anti-protist, you name a biological activity for a molecule, and Streptomyces will probably make something analogous to that.
So you've worked with Mark Charette, and he actually refers to many microbes as being supreme chemical engineers, and this is a great example of that kind of thing. They are really quite amazing, and I love the fact of how hydrophobic the tops of the colonies are. I mean, I personally have dropped water on them and seen it beat up. And if you think about it, that really helps the spores move around when things are wet.
Yeah, I mean, if you actually drop into a really mature sporulating colony, which you can see on the extreme right of those colonies where they go then grey as the spores mature, if you drop water droplets onto a lawn of these organisms and you roll them around, you can actually start to see the powdery appearance of the spores sticking to the outer surface of the droplets. That's literally how we harvest the spores in the lab. We add that, and then we use cotton buds, which I guess in the US you guys would call Q-tips, and we just rub them over the surface, and the spores come off the mycelium. And we filter them through cotton wool to get rid of the hyphae that might come off as we rub them, and we have a really nice dense spore stock that we can use to inoculate cultures from.
What I like is how much we know about the organisms initially because of some of the chemicals that they produce. If I get to teach microbiology again, I'm certainly going to create a slide of all the different things that the relatives of Streptomyces actually make. And there's lots of them, as you mentioned, ranging from immunosuppressants. Isn't rapamycin also from an actino?
Yeah, rapamycin is from a Streptomyces as well. So yeah, it's kind of interesting because I always ask my students this is why do you think an organism might be making an immunosuppressive? They make anti-cancer drugs. I mean, there's not free living immune systems and cancer cells living in the soil. And of course, most of these are altering gene expression in things like protists. And I mean, what is an amoeba if not a free living macrophage in the soil? So they're actually attacking these organisms and altering these organisms' behavior in order to stop them being predated. And we can exploit these for modern medicine. And of course, when Waxman was inspired by Fleming's work to start to look in the soil for organisms that made bioactive molecules, he hit upon the Streptomyces pretty quickly, discovering streptomycin from Streptomyces griseus. And that really kicked off the kind of what we call the golden age of antibiotic discovery in the 50s and 60s, which is well known. And all of our drugs that we commonly know as antibiotics from these organisms were isolated in that kind of 25, 30 year period up to the start of the 80s.
So it's wonderful for students to see those zones of inhibition from isolates, which I use with the Tiny Earth program that Joe Handelsman put up. But also I was fortunate enough to get to know Julian Davies a little bit, and he had so many stories about the early days of antibiotic work. And I'm going to put some links to those things in here.
Jules was amazing. Yeah, I really miss him. Sadly, we lost him fairly recently.
Yeah.
But yeah, a really humane person, very funny, always had a really good story to tell.
He did.
And yeah, just one of the kindest, most generous microbiologists I think I've ever come across. He was always very kind to me, and yeah, and he still continued to play rugby up until his 60s, apparently.
Well, all I know is that when he was in his 80s, when I met him, maybe early 90s, he was much sharper than I've ever been. So there you go.
I think Jules was much sharper than most people.
Yeah.
I just remember being at a meeting in Prague, oh God, maybe 2006. And Julian was saying he'd been chatting to some students and they'd said, "Oh, it's really amazing to meet you. I don't think I've met a microbiologist as old as you." And he said, "Oh, hang on a minute. I'll introduce you to Arnie Domain, who was in the same meeting."
That sounds like him. So for listeners and viewers, it's important to know that the antibiotic revolution that we take for granted was created by these folks like Waxman, Julian Davies, taking you back to Fleming, though that was a little bit different. The thing to say is that revolutionized our world. And so we are faced with something. The fact is that there are many mechanisms that bacteria that cause disease can use to overcome the effects of these antibiotics. Now, I will say that these antibiotics have taught us a lot about how certain aspects of the central dogma of molecular biology work. I mean, streptomycin affecting the way that protein synthesis works in a very specific fashion, tetracycline in a different way, etc. All of these from Streptomyces, by the way, which is fascinating to me. I've had students ask me many times if the microbes that make antibiotics are using them to keep other bacteria at a distance in the soil. And I don't know if people have been able to study that because of the range of things that you have to look at. What are your thoughts on that?
So this has always been the kind of accepted dogma within the field, is that they initiate development because they're stressed and they're running out of nutrients, and they initiate production of these bioactive molecules at that point to protect the remaining resources available to them. So either killing or keeping away organisms that may utilize the last of those resources. The evidence to support that is actually very, very slim, because those are very challenging experiments to do technically. In the last kind of 15 years or so, there's been some really interesting work that's come out of both students and postdocs with Roberto Colter. So Paul Strait, who's now at Texas A&M, who's an amazing microbiologist, and also Matt Traxler, who's in Berkeley, who's also doing some fantastic work. And what they were able to show is that if you grow Streptomyces next to colonies of Bacillus, you can get them to turn on antibiotics that they don't normally make in the laboratory. So there's something that they're sensing produced by the unicellular organism that switches on the so-called silent biosynthetic clusters that they have in their genomes. There are some really beautiful images where you can apply things like imaging mass spectrometry, so you can scan a laser over the colonies and you can identify the molecules that are volatilized from the surface of the colony. And you get some really pretty pictures in those images, and people have discovered all sorts of molecules that way.
So what's interesting to me is that I was very influenced by Julian Davies, though I could have never called him Jules. He was very nice to me. He was like an elf. You know what I mean? He really was. He was this powerhouse, petite powerhouse of energy. But he had a strong feeling that antibiotics were more like signal molecules, which is something that you're referring to here. Because when we use them clinically, we're using them at much higher concentrations that would ever be seen in nature. And so I'm a little bit prejudiced in favor of that, because I became I didn't become, but I was very influenced by Julian Davies. I'd hardly call myself an acolyte. But I think it's absolutely true. And he was able to show that particular genes were turned on by very low concentrations of antibiotics. And that could be part of it. What we don't understand, and this is why I love the term socio-microbiology, we don't understand interactions very well because we look at things by themselves. And also because we found out that many of these organisms make things that could be of medical relevance that's where we're going to put our attention. And that's why the pure research is so important, because it tells us what's behind the story.
Oh, absolutely. I mean, you alluded to it before about how much basic science has been yielded from studying molecules, and a lot of our chemical biology tools that we use have come from natural products that were never really suitable for a whole range of reasons in the clinic. One that springs to mind is globomycin, made by Streptomyces globosus. And that actually targets lipoprotein maturation. So people who work on lipoproteins often use globomycin to switch off the cleavage of the peptide during lipoprotein maturation, and it gives you a good indication of whether it is a bona fide lipoprotein. But there are lots of examples out there of molecules, and I think Julian was right on his idea about the parvome and how these were signaling molecules. And it kind of fits, is that many of the molecules that are made from these enormous biosynthetic gene clusters, you actually don't get a single molecule. You get a complex of slightly different decorated molecules that can influence organisms in a different way.
I don't want to brag, but you do realize that I coined that term for Julian, parvome.
Brilliant.
Well, he said to me when I was meeting with him, he was in British Columbia at UBC, and he said, "I wish there was an omics for me." And I said, the Latin for, and especially when there are small molecules. And I said, well, I think that's really important. And the Latin for small is parvus. So why not the parvome? And I just love the fact that it's become something that a lot of folks have talked about, because it's really true. We tend to focus on like big title things, and we don't look at what takes place beyond. And so I'm delighted to hear you use the term.
Well, it's great. And the late, great Roger Hendricks would have said, everybody needs an ohm of their own.
Yeah, Jonathan Eisen often has like the bad omics awards, and I understand it can get overused, but it's science is a human profession, and we have all these kinds of aspects to it. And most importantly, just because we want something to be true, nature doesn't care.
Absolutely.
I got told this by Bill Metcalf many years ago, that phenotypic assays in microbiology are for your benefit, not for the microbes.
Very true.
And I have been fooled many times by a phenotypic assay. So you learn. And so this is what's so exciting when you start with this organism that was very important for medical purposes, and then folks got interested in how it was able to do these things. Some of that translates to improvements, but others to basic knowledge that can be applied elsewhere. But looking at it the way that you just described, where you can actually see how these organisms are existing in the soil, is very important. Jo Handelsman doesn't like the term dirt because she feels it's disrespectful, and it's an ecosystem, right? And it really is.
Yeah, I mean, I think we also, even if you use soil, I mean, of course, soil is bulk soil, and within even a square centimeter of soil, there are lots of different ecological niches within that soil, and we need to think about this on the micro scale, not on the kind of millimeter or the meter scale. And I often show my undergraduates a picture of the Serengeti and say, right, okay, so you can see zebras, you can see lions, you can see trees and grasses, but of course, if we look at a gram of soil, or actually the thing is 10 grams of soil, there's more biodiversity in that than there is on the entire planet of macro diversity. And I've known that fact for probably 15 years, and it still blows my mind.
So a million years ago, when the dinosaurs ruled the earth and I was taking microbiology at UCLA, Sid Rittenberg was my first microbiology professor. It's what got me interested in Della Vibrio, actually. But he used to tell me that you could take a teaspoon of soil and isolate everything that we're looking at in microlabs. And you can take that too far, but it's absolutely true that there's a huge amount of diversity in there. And it really surprises the heck out of students when we actually do platings. And then I tell them, and these are just the ones that will grow on nutrient media.
Absolutely.
I mean, it's absolutely as complex as everything else.
Yeah, I think it's really difficult to kind of comprehend, even for us, the number of organisms that are in 10 grams of soil on the planet at any one time. And I mean, I can't remember the figures now. It's like 10 to the 30 bacterial cells on the planet at any one time. And an order of magnitude larger for phage on the planet at any one time. Yet astronomers account for 10 to the 22 stars in the entire universe. So you're talking multiple orders of magnitude larger, and I don't think humans can really get their brains around those kinds of numbers.
So during the last podcast that we were talking about marine microbiology and picocyanobacteria, and I actually put a link up to a paper from Nature called Microbiology by the Numbers, and I'll probably put it in the show notes for this as well. And it deals with exactly this issue. And it kind of takes me back to JBS Haldane, who said the universe is not stranger than we imagine, but stranger than we can imagine. And I think that level of diversity and number are really hard to bring across to people.
I mean, I mentioned Roger Hendricks a little bit earlier, but Roger always used to say when he gave his phage talks was that, and it's another Haldane quote, is that if there was a creator, that he would have an inordinate fondness for beetles, because at the time there were 300,000 validly described beetle species. And of course, Roger would say he would have had an inordinate fondness for phage.
Yeah. I couldn't agree more. It's so interesting. And it's like the old philosophical thing. It's turtles all the way down. I often say that, in fact, I don't know if you can see it, but yeah, it's right here. I like tardigrades very much. And an artist friend of mine did tardigrades on top of tardigrades on top of tardigrades based on that. But it's microbes all the way down. Let's face it.
Yeah, absolutely.
But no, this is very important for people to see, because sometimes we get focused on applied research, and this is really good. I mean, I have never done anything that benefits humankind, okay, in the lab, ever. So let's be clear on that. So people that decide they want to research and do studies on things that have direct application, that's wonderful. I'm not putting it down at all. But pure research actually helps with these kinds of things. So for example, you learn that a lot of the Streptomyces-like organisms tend to make antibiotics at a particular stage of their life cycle. So understanding, how that works can potentially help with the production of more of those antibiotics. And again, Julian Davies had such fun stories about that. But I would go further. You have a beautiful photograph, if I may ask, where someone paints with actinorodin. And you might want to talk about that a little bit, because art really helps bring microbiology to life.
Absolutely. Yeah, I mean, that's very true, is that the molecules that these organisms make and the developmental process are intimately linked, and there's actually quite a lot of regulatory proteins that underpin both processes. There's lots of plasticity in how these organisms express these genes. But the image that you're talking about was done by a former grad student in my lab called Jana, and we had a collaboration with a really, really great friend of mine called Paco Barona Gomez when he was based in Mexico. He's now based at the University of Leiden in the Netherlands. And we were working on trying to understand the kind of wealth of primary metabolic genes that were involved in Streptomyces metabolism, and try and identify genes that were co-opted into biosynthetic clusters, because all of the building blocks to make antibiotics are taken from primary metabolism in order to build those molecules. So there was a competition in Scotland for images of research, and Jana decided that nothing illustrated collaboration than painting Streptomyces map of the world, and then using the mutants that we created that overexpress the antibiotics to highlight us in Scotland and Paco in Mexico. So you can also see the chemical structure of the molecules. So it's a beautiful image that Jana created. I actually have a maybe 3 foot by 2 foot print of it up in my office in work.
No, I mean, and I will tell you that it does such good because so many people have a negative view of microbes in general. And so when I use photobacterium to do art, uh, I know people who kind of look down their noses about it, and they are welcome to do that. It's not the first time I've had people criticize me. But it also lets people who aren't necessarily microbiologists understand that the majority of microbes really could care less about us. I mean, I have a saying I've had for many years. I learned it. I came up with it with Abigail Salyers so many years ago. Is first evolved, last extinct. That's the microbial world.
No, I mean it's absolutely true. And talking of art, is that one of my closest friends in science, a guy called Matt Hutchings at the John Innes Centre. So we were postdocs together, and he was in a pretty successful band. They had a few sessions on the John Peel show on Radio 1. And their first album has the name of the band, which is Bearsuit, written in Streptomyces on the inside of the album cover.
Oh, you've got to send me that image. I've got to
Try and
have
dig
that.
it out. I have to dig through and look for the CD. It's in my garage somewhere. Nobody
Oh, my
plays
goodness.
CDs anymore. So
Have you noticed? Yeah, it's like that. So getting back to the point, we've talked about the life cycle. Now, did you want to talk a little bit about how the antibiotics are made?
yeah. So there's kind of an interesting kind of evolutionary problem with this, is that when Streptomyces grow and they initiate the developmental cycle and they switch on the biosynthetic clusters, these are often very large, complex molecules. So for a molecule like tetracycline, there are about 70 enzymatic reactions required to make that molecule. It's a very highly reduced molecule. So it requires lots of reducing power derived from primary metabolism. For those people obsessed with metabolism like me, there's a lot of NADPH drawn from the pentose phosphate pathway to generate that reducing power. But the biosynthesis of these molecules is from a cluster of genes that we term a biosynthetic gene cluster. And this can be anything from 15 to 20 kilobases for a kind of standard, if there's such a thing as a standard antibiotic, up to 60, 70, 80 kilobases for some of the more complex molecules. And they're just incredible. And of course, as metabolic pathways for primary metabolism have evolved, as genes get co-opted into these pathways, they co-locate through evolutionary processes. And these biosynthetic gene clusters have all of the genes required for biosynthesis. They also have regulatory genes. They have resistance genes, because you don't obviously want to make something that kills you as soon as you make it. Often those genes coincide with the export genes, so they'll have an efflux pump for sending the antibiotic outside of the cell. But sometimes there are genes that are dedicated to modifying things like the ribosome to ensure that you don't become self-toxic by producing the antibiotics. And yeah, many of the genes for modifying the building blocks are duplicated genes, or genes that have been acquired horizontally that actually fit into primary metabolism. So it's a good way of actually identifying unusual biosynthetic clusters that don't really stand out, is that you can look for duplicate enzymes from primary metabolism and then use them to look around the surrounding genes to see whether it encodes a biosynthetic gene cluster.
Yeah, I always get nervous when I see a gene cluster of how there were rodents of unusual size in the Princess Bride. These are gene clusters of unusual size, and when they get together, I instantly think about horizontal gene transfer, right? And I think about genomic islands and all that kind of stuff. So it's just such interesting work to think about, because this is clearly something where the organism is spending a lot of energy. And mostly microbes are fairly conservative about this. I'm not making, that's kind of a judgment, but you understand where I'm getting at. So if they have a bunch of this stuff, there is a use to it, at least under some conditions. Otherwise, why replicate the DNA? But it's a good reminder that these are things that are naturally taking place. It's like a huge chemistry department in the soil.
Yeah, no, absolutely. And I think all of these different functions, I mean, when the first Streptomyces genome was sequenced, which was Streptomyces silicola back in 2002, we knew that it made a blue antibiotic that you can see on that slide I showed earlier. It makes a red antibiotic, which is often confined to the cell, and a calcium-dependent antibiotic. And we didn't really know anything else about the biosynthetic capability. And when the genome was sequenced, you could actually see biosynthetic gene clusters for a whole range of different molecules. At least one other antibiotic encoded in the genome, multitude of siderophores, which are specialized molecules for capturing iron from the environment, and a whole bunch of molecules that we still don't really know the function for, and they're not made under standard lab conditions. That strain also has an antibiotic that is encoded on a plasmid. So we normally think about antibiotic resistance being transferred by plasmids, but these organisms are sending antibiotics to other organisms on plasmids. And methylamycin is made on a large, again, another linear plasmid in Streptomyces. So they have all of these complexities of managing multiple linear molecules within their cells. So they're really crazy organisms, and yeah, they're just wonderful.
So we have this saying at the last microbiology class I talked about microbiology as fundamental, and it's really true. So many students that I get, if you ask them what a plasmid is, they always say small, they always say circular, because that's what the introductory textbooks say, but it's not like that. And what I tell students is this is like when you learn Mendelian genetics. Here's Mendel's first law and second law, and here are the exceptions. And, we like to stick with simplistic things, but the fact is it's going to be as complex and diverse as nature itself.
Oh, I completely agree. I mean, one of the Streptomyces strains we work a lot on is the clavulanic acid producing strain, Streptomyces clavigerus. That has a relatively small genome for a Streptomyces at about 6 megabases, but it also contains a 1.8 megabase plasmid that's linear. It encodes 15 or 20 biosynthetic gene clusters on that one plasmid. And it has 3 other plasmids that are between 11 and 450 kilobases. So it has plasmids bigger than free living organisms' genomes. It's insane.
And I remember when we first started working with the rhizobium megaplasmids, and I said, why not call them mini chromosomes, or a quarter the size of the chromosome? And semantics aside, it just reminds you that, and you used the term, it's one that I've used a lot in my classes, microbial genetics, microbial biochemistry is extremely plastic. And it's all about like Darwinoing things, and it's the hand of Darwin on all things taking place. There's so much going on. Sometimes I worry that I teach students things are too variable.
I think plasticity is something that we really overlook in microbiology. We appreciate it and say, oh, it does this on this medium and it does that on another medium, but we never describe it as plasticity. And I think it would allow us to draw microbiology much more to the ideas that are pervasive in evolutionary microbiology, and would allow us to be a bit more free thinking about why things do things in the way that they do them, if that doesn't sound kind of crazy.
Well, I think honestly, the fact that we really want there to be simple rules, and we want them to be predictable and consistent. And so in the early days of microbiology, go back to Biernik, he would isolate things from nature, and the phenotype he was interested in would not be stable, and he would discard those because they're hard to study. And I hate to use a numerical approach to this, but my guess is there are many types of characteristics of microbes that are variable that we don't think about at all, because we're interested in things that are reproducible and relatively simple comparatively to study, which is why all the improvements in technology are so important, because now we can start to look at those things.
We quite often we will Streptomyces is very plastic. It's very difficult to make it grow reproducibly and do the same things, and it's a challenge for the whole field. And I'll be in my lab meetings with my team, and they'll say, "Oh yeah, I did this, and it doesn't look the same as it did," and we'll all kind of shrug and go, "Streptomyces." And then you can repeat it, and it'll do something completely different. And often, I think, especially when you make a mutant, you see the phenotype will disappear as it picks up compensatory mutations across the genome to allow it to then deal with the medium conditions you grow it under.
Go ahead.
I think the other thing that we think about, if you think about fitness of an organism in a kind of fitness landscape in the kind of classical Sewell Wright type way, we tend to just think of one or two fitness like growth or antibiotic production within that genome space. And of course, as you account for one phenotype, as you add more and more phenotypes, that curve of how reproducible the phenotype is will flatten out across, and especially when you've got 7,825 genes, there's a lot of variation that can happen in that genome.
I think it's important for listeners and viewers to understand. I mean, I keep saying I'm old because it's true. I was trained as a microbial geneticist. And so the idea is I wanted to knock out a gene and see what the effect on a given phenotype was. I knew perfectly well that there were many phenotypes that involved many different genes, but I didn't know how I could study them. But in many bacterial systems, let's just say, for example, I start with an organism that can swim through the soft medium, and I find the fastest ones out of it. And I take that cycle through cycle through cycle. Then I end up after 20 or 30 cycles of something that's much faster at swimming. Now, for about $80, I can find out, if the organism I started with is well known, what genes among all of its genes have changed. And then it's easier to figure out which ones have to do with the change in phenotype that I observe. I never dreamed we could do things like this.
No, I mean, I'm equally old. But I remember being a final year undergraduate when the first free living complete genome of a bacterium came out. And I didn't realize within kind of 25 years, I would be doing those things in my lab for a fraction of the cost. I think when David Hopwood got the funding to sequence the first Streptomyces genome, it cost about £1.5 million. Mm-hmm. We do them routinely for about £60-70.
Yep.
It's peanuts. So now we routinely sequence mutants, and it allows you to go back to the old days of doing random mutagenesis and screening and just looking for mutations in those genomes, rather than checking your mutant by PCR and kind of putting your fingers in your ears and saying la 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 and trying to ignore any of the other suppressor mutations that may crop up.
Well, I used to say, if you make a mutant, you've got to clean it up. You've got to move it to a new organism because there may be other mutations you're unaware of. Even if you're doing transposon mutagenesis, you have to be so careful. And now you don't really have to worry quite so much. I mean, of course you should, but you don't have to worry quite so much anymore. And this is because of the advances in technology. And this is why it's important for students to understand that data analysis, being able to do bioinformatics, which with a small b as opposed to an uppercase B, is a really important set of skills to have, because it's, as you say, getting the data is now cheap. Now what's expensive are the people analyzing it.
Oh, yeah. I mean, we have a long-term evolution experiment running in the lab, and we have loads of data that we just don't have the time, the man hours to analyze. And it would be great to kind of chase every avenue down within that experiment, but we have to be a little bit conservative and choose them carefully, which we follow up.
This is a good segue into something that I've always told students, is that no matter what you do with microbes, natural selection is taking place. The hand of Darwin is on that particular experiment. So I noticed you've done some work with in-lab evolution of traits. Would you care to talk about that some?
Yeah, so we I've I've always loved Rich Lenski's long-term evolution experiment in E. coli. I think I first discovered it when I was an undergraduate and just thought of all of the kinds of possibilities of doing this, and how you could study evolution. And of course, Rich has made an amazing set of discoveries from that experiment. And I always wanted to imitate that, but with our organism. And Streptomyces is fundamentally different from E. coli. Its genome's twice the size. It's linear. It has a developmental cycle, and it makes antibiotics. So we took a little bit of a different angle on this. And I think in the show notes, I think you have a copy of the preprint that we have up in bioRxiv at the moment. And what we did was we took a slight angle on this. So we took a strain where the main antibiotic biosynthetic clusters had been knocked out. And what we do is we passage it in liquid medium, and then periodically we put the biosynthetic cluster back in and ask the question, do you still regulate your antibiotic the same as you did at the start of the experiment? So we can really study epistasis in antibiotic production. Now, if you do that with the cluster present, what you tend to find is most of the mutations that affect antibiotic production map to the biosynthetic cluster. It's the kind of experiment people have been doing for 50 years, started by David Hopwood. So that's one angle. And then also, because the strain we use doesn't sporulate in liquid culture, we can also understand adaptation to growing in liquid rather than in a structured soil environment. And the answer is that they get really good at growing in liquid culture, and they abandon quite a lot of their developmental life cycle. So we get lots of mutants that we refer to as bald mutants, which don't make aerial hyphae. And we get a lot of white mutants, like the ones in the center, which don't go on to fully complete their developmental cycle. What we then have to do is we've sequenced the genomes, we've sequenced the populations, but is going back and then really digging down into gene function. We have some really interesting developmental mutants that there are no mutations in any known developmental genes. So we can really start to really dig down into the minutiae of how development is regulated.
It's interesting to think about here, because another time I'd like to share with you some of the stuff that I try on my level to get your insights into it, because I think you'll find it interesting. But we have an inability to perceive things microbially, and that's okay. Like Barbara McClintock, a feeling for the organism. We have to have a similar way of looking at the microbial world. And I say this with great respect to evolutionary microbiologists, that I don't think we understand selection as well as we think we do. And also the fact that you can have, what percentage of genes in E. coli have no known function? And there's quite a few, but they're still necessary. We just don't know what it is they do. And this issue of interactivity happens. And I would imagine that there are a lot of emergent properties between things that don't seem like they would work together very well, but actually do in vivo, because we're reductionist in the way that we look at things. And that's paid off a lot. But now it's time to look at interactions. And I think that's what you're dealing with there, is finding new functions for genes.
Yeah, I think we certainly have a lot of mutations and deletions in genes of unknown function. I think the figure is something like 30 or 40% for all sequenced bacterial genomes in terms of the genes of unknown function that they contain. And that number's not really dropped in the 25 years or so that we've been sequencing bacterial genomes. And I think we've kind of unearthed quite a lot of the genes that are easy to find functions for. And now we're looking at genes where you might require a synthetic lethal, so that ability to make a knockout in one gene and then make a parallel knockout and then find the gene function. And of course, the one that always jumps into my mind, because I teach it to my undergraduates, is the chaperone protein skip and SARE in E. coli as a synthetic lethal. But there are hundreds of these out there. Problem we have in Streptomyces is our genetic tools are probably about 25 years behind E. coli. So we have CRISPR systems now, but they don't function in every strain very well.
Right.
There's a lot of variation. We still make a lot of our knockouts in cosmids and then make the cosmids from E. coli into Streptomyces and look for double crossovers. So it takes a long time. I'm always envious of my colleagues who work in bacillus who do a PCR, take that PCR product and drip it into a culture, plate it out later that day, and the next morning they have the mutant. If everything goes swimmingly for us, we can make a mutant in about a month.
Yeah. And this is the thing that I tell students that go on to graduate school is picking your system's important, but you still have to follow, what's the saying? The microbial heart wants what the microbial heart wants. And for me, I've always liked weird microbes, undomesticated microbes. And I don't know why, but I always have, because it doesn't make sense. You want to see something that's really well understood. Depends on the questions you're asking, though.
Yeah, no, absolutely. And I mean, Streptomyces just look and smell so beautiful. So why would you not want to work on them? No matter how much of a pain they are.
I'm wearing a pin here that says I have to say weird stuff or I'll die. And that's 100% who I am and will always be. And what I love about this is you're talking about the fact that we can use evolution in the lab, which, again, a lot of us knew. We really respect the heck out of Rich and his work, and he's a fabulous human being, by the way.
Yeah.
But in addition to all of that, this is something that we can now do in high schools. And it's a wonderful idea to start having this trickle down into the way that we look at problems, because just as the physicists used to say that physics advances via gravestones, which is a terrible thing, but also has truth, I would argue that actually a field advances because of how you teach early on. And there's too much hoop jumping and not enough investigation, to my mind.
Yeah, no, I would completely agree. I would also agree that Rich is one of the best human beings around. He's just a fantastic guy. Last year, he won a prize at the Microbiology Society, so I was lucky enough to have dinner with him a couple of nights, and the time he spent with my students and postdocs talking about our experiments and some of the ideas that he gave them was just priceless. So generous with his time.
He gets unhappy with me when I say this, but I think it merits saying. When I wanted to move back to academia from biotech, I wrote him a fan letter. And he interviewed me for a job. I didn't get the job, but it was one of the best times I've ever had. I have never been turned down for a job more kindly, productively, and humanely than by Rich Lenski.
Yeah, no, super guy. Really friendly and engaging, and his prize talk was just magnificent. I think you can probably get it on the Microbiology Society website.
I think I'm going to put a link to that, because I think that's a lovely idea. I
mean, we got into the long-term evolution experiment with Streptomyces through trying to understand how lineages of industrial Streptomyces have been developed. So we're very lucky. We have a long-term collaboration with GSK, who make clavulanic acid about 30 miles away from my lab. And they've been kind enough to share part of their early lineage of Streptomyces claviligerus with us. And we've been sequencing genomes and trying to piece together how they have evolved in order to understand why they've become good at making lots of clavulanic acid, and also how they've evolved to grow in a fermenter. So one of the things that always gets leveled at you when you're an evolutionary biologist who works in the lab is that they say, well, it's all very well growing your organism, but soil is not like agar. But of course, we have the argument that we're not trying to understand how these organisms grow in soil. We're trying to understand how they grow in the lab and how they evolve and adapt to make antibiotics for human use.
As long as you have the original strain with which to compare it, and that's what's so important about it.
And the niche that they're adapting to is the fermenter.
I got to say, I'm sure if you did things from plate to plate versus flask to flask, you would get very different paths. And it makes perfect sense why.
So Danny Rosen in Leiden, who's a brilliant evolutionary microbiologist, has done some really wonderful Streptomyces work on division of labor. And they've done a lot of their work trying to look at how sporulation is involved in antibiotic production and that regulation. So Danny's work is really worth a look as well.
Given the problems that we face with the antimicrobial resistant problem that's coming down the pike toward us, where many disease-causing organisms become resistant to all known antibiotics, what role do you think your studies in Streptomyces can help us in overcoming that?
The problem with antimicrobial resistance in the clinic is absolutely an evolutionary biology problem. So whatever antibiotics we discover and bring to the clinic, there will be resistance within a short period of time. And this is partly where the problems lie, in that the pipeline for new antibiotics to reach the clinic is actually very, very dry. It's almost at a drip where very few molecules are coming to market. So we need to continue to discover new organisms. We need to mine their genomes for these biosynthetic clusters. And of course, we mine their genomes looking for biosynthetic clusters that we know, because it's very difficult to look for things that you don't know. We had a paper in MBio a few years ago, the known unknowns, the kind of Donald Rumsfeld biosynthetic gene cluster that kind of got quite a bit of attention, with a lot of teasing from my friends. But the issue we have really is, if you can discover new molecules and you can activate them or move them into a synthetic biology chassis and express them in the lab, then that's great. But I think the power of modern DNA synthesis and molecular genetics means that we can engineer biosynthetic clusters. And this is being done where you can introduce different enzymes and decorate the molecule in a slightly different way. In the early days of this, David Hopwood's group with Paco Malpartida showed that if you mix two polyketide biosynthetic clusters in the same strain, they make a novel molecule that doesn't exist in nature. So this ability to make, I think what David calls non-natural natural products is an opportunity that we have. And I think with all of that biosynthetic capability and blueprints that are out there in the genomes of these organisms, we just really need to start doing the experiments and mapping them. So yes, I think we can make and discover new molecules, and some that don't exist in nature, but we'll never discover an evolution-proof drug that won't ultimately discover resistance in the clinic.
I think that's so well put. And a lot of times it's because of the Red Queen issue. So one half of the equation doesn't change and one does. So that one's going to eventually overcome it. I think Fleming was able to show resistance to penicillin really quickly. And
I think Fleming showed that before penicillin was even in the clinic, that you could get what he referred to as penicillinases, which we now know we call them beta-lactamases. But yeah, they were around way before. Yeah. And many of these organisms are producing them because they are using them in the natural environment to compete with these organisms. One caveat to all of that is that many of the resistance genes that we see in the clinic are not the kinds of resistance genes that we see in the biosynthetic gene clusters.
That's very interesting. I did want to say, by the way, that's the value of programs like Joe Handelsman's Tiny Earth, the idea of using students to look for antimicrobial producers and create kind of a repository for them, because as you've mentioned, most of the pharmaceutical concerns are not actively looking the way they used to. And I think we need to do everything.
Yes, no, I completely agree. This is the kind of impending pandemic that nobody really wants to talk about, because it's not sweeping and affecting every single human on the planet at the same time, but it will affect us at some point.
I agree. Getting back to data analysis, I had a couple of questions about that. So do you think that AI approaches could help you with unraveling some of these biosynthetic gene clusters?
So there have actually been a couple of recent papers where people have used AI to design new molecules and discover new molecules that are or have applications as antibiotics. So people are already doing this. It's way beyond my skill set. I'm quite happy to stick with molecular genetics and screening and bioinformatics. But yeah, computational mining and quantum computing for these kinds of things needs chemists and computer scientists, not old people like me.
I feel you, as the kiddos say. But what I would point out with all of this is what happened when we started analyzing DNA sequence data is that the people who collected the data weren't so good at understanding how to interpret the data, so folks had to work together. So I imagine for issues like AI or the burgeoning field of quantum computing, we're going to have to learn to speak a similar language. And I think that's all to the good.
I think that's we've worked a lot with chemists and physicists and engineers over the years on a whole range of multidisciplinary projects. And actually, most of it is just learning each other's language.
Yes.
And if you understand each other's language, then you can then start to make progress. So I think collaboration is the way to go.
Yeah, I find a lot of people don't appreciate that, or they'll be negative about it. They'll say that no one person understands all of it, to which I say, yeah, but it's implicit on everyone in the group to have that common language so that we understand what's taking place. When I annotate something, I'm using a computer program to annotate it. Am I a computer scientist? I am not, right? But what I do like to do is experiments based on that.
Completely agree. 100%.
This has been a really fabulous session, I have to tell you, Paul. I so appreciate your time today. I've learned a lot. You're as enthusiastic as I am, so that's always good to see. And I think listeners and viewers will get a lot out of this. But before we go, what would there be 3 things that you'd like people to remember, and why isn't frogs one of them?
One, Streptomyces are absolutely amazing organisms. I think their biosynthetic capability, as take-home message 2, is vast, and we've just scraped the surface of really what this biosynthetic capability is within these organisms. And I think we know very little about their evolutionary interactions with other organisms, and I think that is going to be where the next big discoveries in Streptomyces biology are. People like Matt Hutchings, I mentioned before, Mark Chevret, who you mentioned, and Cameron Curry have done some amazing work on the Streptomyces interactions with leaf-cutting ants.
Yep.
And the work of Paul Straight and Matt Traxler in terms of interactions with other bacteria. We've done a little bit of work where they interact with amoeba, and I think there is just so many potential interactions that will activate biosynthetic clusters, will allow us to discover new biology that could be broadly applicable across a lot of soil organisms.
I think that this is a really good example of the connection between industrial microbiology, clinical microbiology, and microbial sciences. Absolutely. And it's been a wonderful session, and I want to wish you and yours the very best, and I so appreciate your time for today.
And we didn't even talk about frogs.
Thanks so much.
Absolute pleasure, Mark. Thanks for having me.
This has been Matters Microbial, a podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as always with epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Professor Paul Hoskisson can be found in the Strathclyde Institute of Pharmacy and Biomedical Sciences at the University of Strathclyde and Glasgow, Scotland. Many thanks, as always, to David Renata for superb and very patient editing, and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.